Hi there. Have you ever asked yourself if sickle cell disease can truly be cured? Well, today we are diving into that very question, and guess what? The answer is yes. Sickle cell can be cured. Let's explore the two advanced treatments that offer real hope for patients with the condition. Before we dive into these treatments, let's get familiar with some key terms. Have you ever heard of stem cells? What exactly are stem cells? Well, stem cells are special cells in the body that can divide and develop into different types of cells. For example, we have blood stem cells, known as hematopoietic stem cells. These stem cells reside in the bone marrow and are responsible for producing all types of blood cells, including red blood cells. They carry several important genes that control the production of different blood cell types and their components. Some of these important genes include the beta globin, gamma globin, and the BCL11A genes. Now, the two advanced treatments that can potentially cure sickle cell disease are bone marrow transplant and gene therapy. For the past decade, the bone marrow transplant has been the only cure for sickle cell disease. However, gene therapy has rapidly advanced and is now generating significant excitement as a promising new treatment. Let's dive into how these treatments work. Previously, we've learned that the defective beta globin gene in sickle cell patients is responsible for producing sickle hemoglobin. And now, we know that these beta globin genes originate from blood stem cells or hematopoietic stem cells in the bone marrow. This brings us to the key role of bone marrow transplant. The aim of a bone marrow transplant is to replace the sickle cell patient's blood stem cells with healthy stem cells from a suitable donor. These donor stem cells contain the correct version of the beta globin gene, and so after the transplant, the new stem cells start producing healthy hemoglobin, which in turn make normal red blood cells. So, what are the steps involved in a bone marrow transplant? Let's take a closer look. In a bone marrow transplant, the defective stem cells in the patient's bone marrow are destroyed or eliminated using chemotherapy, sometimes combined with low-dose radiation. Healthy stem cells with functional beta globin genes are then collected from a donor and transfused into the patient. So the replacement is done just like receiving a blood transfusion. Once these stem cells are infused, they migrate to the bone marrow where they engraft or attach to the marrow and start producing healthy red blood cells containing normal adult hemoglobin. Over time, these new healthy red blood cells circulate throughout the body and the patient stops producing sickle red cells, ultimately becoming free from all related complications. While bone marrow transplant can offer a cure for some individuals, it is generally recommended for severe cases due to the significant risks involved. Unfortunately, not everyone is a suitable candidate for this procedure. Let's explore some of the associated challenges. Finding a compatible or suitable donor can be very challenging, especially if the patient doesn't have a healthy sibling. The chances of finding a donor outside the family decrease significantly, making this a critical hurdle in the bone marrow transplant process. Also, the chemotherapy and radiation used to prepare the patient have side effects. Some patients may experience temporary hair loss, infections, and other complications. However, these can be carefully managed by healthcare professionals. That is a brief overview of bone marrow transplant. Now, let's explore gene therapy. Gene therapy is a groundbreaking medical technique that scientists use to fix or replace defective genes in a patient's stem cell. Unlike bone marrow transplant, gene therapy does not require a donor. The goal here is to modify the patient's own stem cells to correct the genetic mutation responsible for the disease. Let's break it down. In a bone marrow transplant, the the patient's bad or defective stem cells are replaced with healthy ones from a donor. In contrast, gene therapy involves removing, modifying, or editing the patient's own defective stem cells and then reinfusing them into the patient's bone marrow. This sounds straightforward, right? But this procedure is far more complex than it seems. There are three main types of gene therapy. We have gene editing, gene addition, and gene silencing therapy. In gene addition therapy, a functional copy of the beta globin gene is added to the patient's stem cells, enabling them to produce normal hemoglobin. In gene editing therapy, a gene editing tool is used to correct the mutation in the patient's gene. With gene silencing therapy, the defective gene is turned off, allowing the body to use alternative natural mechanisms to manage the condition. Currently, 2 gene therapies have been approved for treating sickle cell disease: Casgevy and Lyfgenia. Casgevy is a type of gene editing therapy, while Lyfgenia is a form of gene addition therapy. Let's take a closer look at how these 2 therapies work to potentially cure sickle cell disease. Casgevy utilizes a gene editing tool known as CRISPR-Cas9 technology. This innovative tool acts like molecular scissors, precisely correcting the defective gene so that it can function properly. In this therapy, hematopoietic stem cells are collected from the patient's bone marrow, and the CRISPR-Cas9 technology is used to edit the BCL11A gene within the stem cells. After editing, the modified stem cells are reintroduced into the patient. This gene modification prevents the BCL11A gene from reducing gamma globin gene activity. Once the BCL gene is successfully edited, it no longer suppresses the production of fetal hemoglobin. As a result, the modified stem cells begin generating high levels of fetal hemoglobin in red blood cells. Fetal hemoglobin does not form sickle cells, so its continuous production improves oxygen delivery and prevents the sickling of red blood cells. This enhances the overall condition of patients with sickle cell disease and alleviates all related complications. Now that we have explored the Casgevy gene editing therapy, let's move on to the gene addition therapy, Lyfgenia. Lyfgenia utilizes a lentiviral vector, which acts like an envelope to deliver genetic material directly to stem cells. The procedure in Lyfgenia therapy is somewhat similar to Casgevy. However, in this treatment, the lentiviral vector delivers or adds a functional copy of the beta globin gene to the patient's hematopoietic stem cells. Afterward, these modified stem cells are transplanted back into the patient. By introducing this healthy beta globin gene, Lyfgenia helps produce healthy adult hemoglobin, which make normal red blood cells. Now, you might wonder, if a new healthy beta globin gene is added, what happens to the defective one? Will it still produce faulty hemoglobin? This is the same question that arises with Casgevy therapy, right? Well, the defective gene remains, but the newly added functional beta globin gene in Lyfgenia and the activated gamma globin genes in Casgevy produce enough healthy hemoglobin to compensate. This means that while the defective gene is still present, its impact is greatly diminished. The functional gene produces enough healthy hemoglobin to reduce the impact of the sickle cells, helping patients overcome the complications they previously faced. Although both Casgevy and Lyfgenia are still largely experimental and require highly specialized medical facilities, they represent major breakthroughs in treating sickle cell disease. These therapies offer hope for long-term relief and a better quality of life for patients with the condition. I'm glad you made it to the end of this educational video. If you enjoyed learning with me, make sure to like, subscribe, and share. Thanks for watching.